Population genetics. It's actually one of the hardest topics on the second half of the year. Some people actually don't follow along very well, but it's not that hard. It's kind of like getting evolution and putting it together with genetics. So it's the bastard child of evolution and genetics put together. And in fact, we call this the modern synthesis theory or the idea of combining or understanding of modern genetics or inheritance, especially with an aspect of a molecular and chromosomal inheritance with the idea of evolution or the concepts of evolution that Darwin discussed. So that's why I have a picture here of the fathers of both things. You have Gregory Mendel and Charles Darwin, which worked together even though they were years apart. By the way, do you know that actually Mendel sent an essay or a letter to Darwin, because Darwin was famous, and they coexisted in time. Towards the end of Darwin's life, Mendel was doing his work, and he said, "Hey, listen, this is my work." But Darwin has so much correspondence that we see from so many people that he kind of put it in the back of his pile, and he never actually got to it. And that's unfortunate because Darwin spent a lot of his time trying to understand the mechanisms that led to the variation that he saw and that he explained through his theory, but he could never actually explain how this variation got passed on from generation to generation, and Mendel ultimately had the answer. But we've come a very long way since both of these two people worked together to create what is now the theory of evolution. So let's talk about some of these advancements that were done. Remember, the idea of population genetics is studying the genetic composition of a population and as it changes over time, which is basically evolution. So the modern synthesis theory is trying to put together genetics and evolution, and the leaders of this were two scientists called Wright and Fisher. And they study how genes vary in the population over time, and they actually study the differences between micro and macro evolution. Microevolution refers to molecular changes or things like mutation or recombination. Remember, recombination is just what happens through sexual reproduction when the genes get shuffled around in a population. And mutation is actual new changes that occur because of molecular changes in the sequence of the DNA code. But together, these two things will create variation in a population. And if these things, there's exposed to natural selection or isolation or other processes which we're going to discuss in this lecture series, it's possible that these things become fixed in the population through things like selection, like drift, and migration. And we're going to talk about each of these concepts of the population. And that's when you get to evolution. And evolutionary hypothesis is trying to predict how fast or how much something changes because of the effects of both individuals and ecology. In other words, how the environmental changes on changes that occurred within the individual. So microevolution are things that start at the organism level with things such as mutations and genetic recombination. So that's everything to do with genetics, of course. But it will also affect the way, depending on the way the animal develops, because sometimes the environment itself will change the way that the animal presents the genes. Remember the concept of epigenetics. And this eventually leads to changes within the population, because those animals which actually have better mutations and developed better will actually become more likely to survive, and it will change the composition of the population. But then it's not might necessarily be evolution in the sense of a new kind of species. It just might be a change within the same species, kind of like why we have different races on the human race. But then with things like migration and dispersal and genetic drift, which we'll talk about, these populations can change even more. And if they get separated over time and enough change takes place, then you start getting into the realm of actual macroevolution. But remember, microevolution includes both changes at the organism level, which include genetical changes and developmental changes, and then changes that involve the interactions between animals at the population level, which include things like selection and migration and genetic drift. But all of these things are called changes within the species. It's not yet a different species. But if there's enough isolation, you start getting independent evolution, which we also call vicariance, when one species is on one side and one species is in the other, and that's going to lead to speciation. And this could happen fast or slow. You have either punctuated equilibrium or gradually change over time. But as then these species get exposed to different historical constraints or developmental constraints, it will lead to what we call species selection or speciation, where one species will be more likely to survive than the other, and then ultimately you get only one of the two sometimes be present in the world. And this will take a very, very long time, and that's what we call macroevolution. Throughout the entire process, it's an interaction what happens within populations and organisms and what happens with the environment, because the environment will constantly changing. The climate, the geology, the ecology of the environment, everything will be changing as the species is changing. And that change relationship between the changes in the animal, the changes in the environment, will cause changes in the pressure, changes in the population, and ultimately changes in the way the life looks. And that's why there's so much variety in life, and both across space and time. And that's what we got to talk about in this lecture series. Some of the concepts that we're going to need to know to understand this include advanced genetic relationships. Now remember, we talked about in the genetics lecture series, we talked about a lot of these things. For example, there's epistasis. And epistasis is when one gene depends on another. For example, if you want to determine the skin color, you're going to need to know, of course, how much color to make. And there's going to be one or many genes which actually decides how much color to make. But there's also going to be a gene that tells you how to make color, period. So it doesn't matter if you tell me how much color to make. If I don't know how to make color, I won't make any. So you see here, for example, the albino mice in this picture here exist because there are certain genes that was mutated, and they happen to have been unlucky in this Punnett square and got landed with a double recessive code for that gene. And although they have genes that tell them to be, brown or black, they end up being white because they don't have the gene that tells them to make the color. So how much color to make and how to make color are two separate genes, but they interact to actually determine your trait. And there's also another cool relationship that we call pleiotropy. And that's when one gene has many jobs in the body. For example, you have one single mutation, but then there's a syndrome that occurs because of that. Think of it, for example, things like sickle cell anemia or Marfan syndrome or Canavan syndrome or so many Tay-Sachs and so many of the other syndromes that we learn about in the chromosomal inheritance lecture series. One gene change can change many things because sometimes that gene is involved in many things in the body. And that's called pleiotropy. Also, there's some genes that have multiple alleles versions. For example, look at blood types. You don't have only the big A or little a, because sometimes when Mendel did his research, he looked at two genetic variations. Only one type of pair could be, only three types of pairs could be created because you can only have those two genes. But that's not actually how it happened in genetics. In fact, the majority of traits you see around have multiple variations of the genes because across the story of evolution, these genes mutate. And so look at blood types. You have the A type, the B type, and the O type, and then even a combination of the A and the B to form the AB type because they're co-dominant together. And so this is what we call multiple allelic characters, such as the blood type, and that's also present when you talk about genetics. There's also epigenetics, which is when genes interact with the environment to actually make the look. And there's no such thing as genetics determining everything or the environment determining everything. The way an animal looks actually depends on a combination of what the environment tells them to do and what the genes tell them to do. And so genetics has got more complicated when you think about the concept of gene expression. If you remember learning about that again in the molecular biology lecture series, sometimes the genes don't get expressed because they don't receive an environmental message for it, or sometimes they get unexpressed or turned off because of what the environment tells them to do. And so this interaction between genetics and the environment leads to more variation within the population even. Even more variation is going to exist because of what we call multifactorial traits, or traits that depend on a lot of different genes put together, like skin color, for example. There's actually several genes which contribute to the formation of skin color. And this is a representation showing with only 3 genes how many variations you can possibly have. But in reality, there's going to be many, many more than that. And that's why there's so many different types of skin color in the population. And it gets very complex when you think about that. When the idea that some traits or characters are determined by the combination of many genes put together. And so it's not just one gene, but it's the additive factor, which you call quantitative factors of all of these traits put together, which will end up determining the actual skin color. Now, that means then that the degree of variation that exists within a population, looking for example at things like hair color, skin color, and all the other things that exist, are going to be a lot because of epistasis, pleiotropy, allelic traits, epigenetics, multifactorial traits. No one is going to ever be the same, even for the same trait. And then imagine, therefore, if you put all the different traits put together, and then you put sexual reproduction on top of that, which jumbles up the genes, the chances of two human beings being sane is going to be very, very small. Which is why, for any given trait, there's a wide range of variation, which we'll call continuous variation between things, usually. Although there are some examples of discontinuous variation in nature, when you have like purple flower or white flower, one or the other. The majority of time, though, you get what is called a spectrum, when you get from one end to a completely different end and everything in between with variation. And that's for each character. Imagine if you put all the characters put together, and you think about that, we are multidimensional levels of variation. We might be the same for eye color, even though the chances of that are minute. But imagine the chances of us being the same for eye color, skin color, intelligence, beauty, height, and everything else. And that's why there's so much variation within the population, and all of this is going to be important in determining whether or not the animal tends to survive or the organism tends to survive and everything. So those are very, very, very important concepts before we get into the idea of population genetics. So I had to review a little bit of genetics because we're going to have to talk about these things. So in population genetics, then, we have to have in mind this idea of genetic variation, that we have a gene pool within a population. The population has several genes from which some animals will take from. Now, maybe some will have these certain genes, some will have other genes, some will have other genes, and depending on the combinations of genes that a specific members of the population have, they may look different from each other. But the gene pool refers to the total number of genes which exist within a population. Look at how much variation, for example, exists in this field of flowers over there. All right? Because different flowers have different genes. Look how much variation exists in the breeds of dogs. Again, because different dogs express different genes. And so that's why there's so much variation. And then there's, of course, the need to understand how common a certain look or a certain genotype or even a certain allele is within the population. And we're going to talk about that on the next video as we talk about how to calculate these genetic frequencies. So we're going to do a little bit of math on the next video. So I'll see you guys then.